to our next speaker, Markus Schosserer, who's been to ARDD a number of times. I don't know how many times you've been here, but I'm very happy that it's just the second time, yeah. But I'm so happy that you are able to speak.
Thank you very much. And also thanks a lot for putting always together this very interesting meeting, and also for giving me the opportunity to present some data from our relatively new lab. And I would like to introduce you into a relatively unexplored topic in geroscience, but which has, in my opinion, still a lot of promise actually. And this is the topic of RNA modifications. RNA modification in general, also called epitranscriptomics, in analogy to the epigenome for DNA modifications. During the pandemic, we performed a very thorough literature search, and we actually found a quite surprisingly large number of enzymes which install RNA modifications and which modulate lifespan of different model organisms, especially with high throughput screens in C. elegans and in yeast. So you see all these green things here are actually enzymes which install RNA modifications and which either shorten the lifespan in most cases because they are either very important or essential, or some of them even extend the lifespan. Almost all RNAs are modified. Maybe mostly studied are mRNAs because it was shown that these modifications are highly dynamic. So they can be added and removed, and they are read by specific enzymes. And which Miller's lab actually showed that some of those enzymes which install mRNA modifications are actually downstream of rapamycin and acarbose, for instance, in mice. However, the most modified RNA species are ribosomal RNAs and tRNAs, and you see especially tRNAs carry a lot of different modifications here in the anticodon loop and especially at the wobble base. But the focus of my talk will be ribosomal RNAs, and on ribosomal RNAs we have in total around 220 modifications in humans. Most of them are either pseudouridinations or ribose methylations, which are catalyzed by either discarine or fibrillary, and which are guided by a SNO RNA. In my lab, we are mostly studying base methylations, which are installed by specific RNA methyltransferases, which do not require SNO RNA for guiding. And especially we look at M5C, so there are only 2 M5C modifications on human ribosomal RNAs. And one of them, so the methylation at cytosine 3782, is installed by an enzyme called Nsun5, sorry. And this modification is localized very close to the inter-subunit surface where the 2 ribosomal subunits come together. Nsun5 is best known because it's highly expressed in many different forms of human cancer, but recent reports also show that it might be associated with RNA virus infections and also ferroptosis. But this is also mostly unexplored yet. And what is really interesting and actually shows the importance is that both Nsun5 and the target sequence on the ribosomal RNA are extremely well conserved between species. So there's a stretch of around 100 nucleotides, which is completely identical between yeast, C. elegans, and humans. We performed some immunofluorescence staining, and we see that Nsun5 is localized right in the center of the nucleoli, so where the ribosomal DNA is transcribed. So therefore we believe that Nsun5 introduces this RNA modification quite early during ribosome biogenesis. And we could also confirm that Nsun5 is really specific for this specific methylation site. So when we knock out Nsun5 in HeLa cells, we see that this methylation is completely gone. But we can restore the methylation by expressing mouse Nsun5. So mouse Nsun5 can completely compensate for the loss of human Nsun5. And now you might ask, okay, what does this have to do with aging? And actually, this goes back to my PhD thesis. And there we found that Nsun5, if we deplete Nsun5 in different model organisms, we extend the lifespan. So first, in C. elegans, if we perform RNAi or a complete knockout, we see an extension of lifespan by around 20%. We see the same in Drosophila. Here the lifespan extension is again around 20%, and we used 3 different RNAi constructs really to show that this is specific. And also in yeast we see an extension of chronological lifespan. What might be the mechanism behind this? So we know that when Nsun5 or RCM1, how is it called in yeast, is depleted, so we completely lose this specific methylation. And then we observed that different messenger RNAs are translated by ribosomes. So in yeast we do not see a specific effect on global translation, but we see specific effects on certain mRNAs. And these are specific mRNAs which are associated with the stress response. So usually mRNAs which are usually activated by H2O2 stress, they are also activated in this Nsun5 knockout yeast cells. So you can think about it a little bit like a hormetic response. And this then indeed also improves stress resistance of yeast cells through both H2O2 and heat stress, and then in the end extends the lifespan in our opinion. And now I would like to share some unpublished data, because of course we wanted to know if this from the more simple model organisms also translates to higher organisms like mice. So what we did is we generated a full Nsun5 knockout mouse model, which has a constitutive Nsun5 knockout in all tissues. I unfortunately cannot yet share lifespan data with you because the experiment is still ongoing, but we already started to measure the clinical frailty index, and there we already got some promising results. So just to remind you, the clinical frailty index, we measure non-invasively 30 different parameters, which give us an indication about the health status of the mice. So if we look at male mice, we see that with age the frailty index increases. This is of course what we expect, and what we were very happy to see is that in Nsun5 knockout animals, which are depicted here in red, we actually see that this increase is slower. So somehow Nsun5 seems to delay the onset of frailty. And this effect was even more pronounced in female mice. So in female mice, you already start to see a clear effect after 9 months of age, and this becomes an even more pronounced later in life. Unfortunately, this improvement of frailty also has some adverse effects. So we also see some trade-offs by the loss of Nsun5, and these include, for instance, that mice have a lower weight, both lean mass and fat mass at all age groups, and both female and male mice. We also saw that Nsun5 might be partially embryonic lethal, because when we cross 2 heterozygous mice, we only get 16% homozygous offspring, where we would of course expect 25%. We also see that Nsun5 loss leads to a slightly reduced bone mass, especially in male animals at around 12 months of age. And finally, this is maybe the most pronounced phenotype, we see that Nsun5 knockout animals show decreased lymphocyte counts. Again, this effect is most prominent in male mice, but you see it's also present in female mice. Now, what might be the mechanism behind this? I told you before that Nsun5 is associated with many human cancers, so therefore we thought Nsun5 might be important for proliferation in more general terms, also in normal tissues. And for this we performed RNA-scope in situ hybridization to detect Nsun5 and Ki67 as a marker for proliferation in kidney and liver tissues, and there we indeed found a clear association between cell proliferation and Nsun5 expression. So most cells which are Ki67 positive or are also positive for Nsun5, while this is not the case for Ki67 negative cells. And since Nsun5 methylates ribosomal RNA, we thought, most obviously the mechanism might be ribosomes, protein synthesis. So we set out to measure global protein synthesis. And we did this first in mouse embryonic fibroblasts, so cells which proliferate, of course, a lot. And there we see a clear decrease in global protein synthesis when Nsun5 is lost. We can rescue this reduction in protein synthesis by re-expression of Nsun5, but not with a catalytic dead mutant. And then we wanted to see if this is also the case in normal tissues. So we injected the mice with OPP and measured global protein synthesis in vivo. And there we actually don't see this effect. So especially in kidney and liver, we see that Nsun5 and wild-type mice are completely identical, while there might just be a very slight effect in lung and in bone marrow. So what we think is going on is we have to distinguish between cells in tissues which are growing slowly and fast-growing cells. So slow-growing cells in somatic tissues or even quiescent cells, they of course have a low protein turnover, and therefore they might not really suffer from Nsun5 loss, and in those cells Nsun5 loss might even be beneficial. While in fast-growing cells like cancer cells, stem cells during development, and then later also mesenchymal stem cells and other progenitor cells, in these cases, those cells have high protein turnover and they might actually suffer from Nsun5 loss. So therefore, when we delete Nsun5, maybe due to the somatic tissues, we see this observed, this delayed frailty, while in fast-growing cells, this might then actually lead to the phenotypes we observed, including body weight loss, reduction in body weight, bone mass, lymphocyte counts, and embryogenesis. Of course, what we are currently testing, what are other possible mechanisms is, again, specific mRNA translation. So what I showed you in yeast, so we are currently testing in mice. And of course, we cannot exclude that Nsun5 also has other functions, like methylation of other targets, or maybe some other functions which are not related to methylations at all. With this, I would like to end here. I would like to announce that I have an open postdoc position in my lab, so if you would like to come to beautiful Vienna, please get in touch with me. And I also wish to thank my current lab, and especially Lisa Lindl and Clemens Heisenberger, who actually produced all of this data which I showed you today. Thank you.
Thank you so much. Great talk, Markus. Really amazing work that you're doing. We have a question from Vera.
Well, amazing work. So what about the fidelity of protein translation? Have you measured it in these mice? Does it affect like accuracy of the ribosome?
Yes, we didn't measure it in mice directly. So what? The results are a little bit different. So when we measure accuracy in yeast cells, we see that stop codons are skipped more often. However, in HeLa cells, we did not observe this at all. So maybe because HeLa cells already have a higher rate of translation misincorporation. So we measured amino acid misincorporation, we measured stop codon read-through, and we measured frame shifting. But in all of those cases, we do not see any effect in HeLa cells. We did not measure the mice yet, but this is definitely on our plan.
Thanks.
All right, we have time for one more question. Otherwise, let's move on. Then we are in good time. Thank you so much, Markus. That was really great. And